Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CUET, that is Central University's Common Entrance Test Question Paper Biochemistry, conducted in 7th June 2023. This is part 2 of the discussion. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of biochemistry, you can refer to my channel playlist. I have uploaded them there. Then next one. The amino acid in the renal tubular cells that form ammonia, which is finally excreted as ammonium, is. So glutamine, it is converted to glutamate by the enzyme glutaminase in renal tubular cells, and this glutamate is deaminated by the enzyme glutamate dehydrogenase to form alpha ketoglutarate and ammonia. The ammonia is excreted as ammonium ions in the urine. So the answer here is option 4, that is glutamine. Rifampicin inhibits, so it inhibits RNA polymerase. So which subunit? It is the beta subunit, and it is the prokaryotic RNA polymerase that is getting inhibited. So option 1, beta subunit of prokaryotic RNA polymerase. Answer is option 1. Then next one. The acid fastness of mycobacteria is due to the presence of mycolic acids in cell wall. Answer is option 1. Then next one. The branch of biology that involves creation of small living organisms like mycoplasma, etc. is called synthetic biology. Answer is option 4. Next one. Which group of the antibiotics inhibit translation by binding to 50S ribosomal subunit? So erythromycin and chloramphenicol, they bind 50S ribosomal subunit. So answer is option 1. Given that delta G is equal to delta H, it is minus T delta S. With reference to it, identify the wrong statement. In exergonic reaction, delta G is negative. Yes, that's correct. When delta G is 0, the system is at equilibrium. That's also correct. Delta G must be positive for the reaction to be spontaneous. No, it should be negative for the reaction to be spontaneous. So the wrong statement is option 3. Then the reaction 1, glucose plus phosphate giving glucose 6-phosphate plus water can occur only when reaction 2, that is ATP giving ADP plus PI occurs. This is because, so reaction 1, that is glucose giving glucose 6-phosphate plus water, it requires energy to proceed. So glucose 2 is exergonic, that is it releases energy. So when reaction 2 occurs, the energy released is used to drive reaction 1, making the overall reaction as glucose plus ATP giving glucose 6-phosphate plus ADP. And here the delta G0, it is negative. This coupled reaction is energetically favorable, allowing glucose to be phosphorylated to glucose 6-phosphate. So the answer here is delta G0 of reaction 1 is positive and reaction 2 is negative. So that is the reason why reaction 1 can occur only when reaction 2 occurs. Then oxidation of one molecule of FADH2 in the respiratory chain forms how many molecules of ATP? So it is 1.5 through complexes 2 and 4. FADH2 enters the respiratory chain at complex 2, not at complex 1. So the energy ATP produced by FADH2 is less. So it is 1.5 through complexes 2 and complex 4. The breakdown of long chain fatty acyl acids to acetyl-CoA via beta oxidation take place in? So it take place in the mitochondrial matrix. Answer is option. Next question. During glycolysis, conversion of 1 mole of fructose 1,6-biphosphate to 2 moles of pyruvate lead to the formation of how many moles of NADH and how many moles of ATP? So they have not started it from glucose, they have asked from fructose 1,6-biphosphate. So it, by action of aldolase, it give rise to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Then by triose phosphate dehydrogenase, you get 1,3-bisphosphoglycerate and 2 NADH. But then 1,3-bisphosphoglycerate is getting converted to 3-phosphoglycerate by phosphoglycerokinase and 2 ATP molecules are produced. Then 3-phosphoglycerate, 2-phosphoglycerate, no ATP and NADH. Then phosphoenol pyruvate, 2-pyruvate by action of pyruvate kinase, again 2 ATP molecules are produced. So how many ATP? 1, 2. So 2 plus 2, 4. 4 ATP and 2 NADH. So the answer here is option 1. 2 moles of NADH and 4 moles of ATP. Then next one. The concentration of OH minus in a solution with H plus concentration of 1.3 into 10 raised to minus 4 molar is. So we know the dissociation constant of water, that is Kw, that is concentration of H plus into OH minus, is 1 in 10 raised to minus 14. So substitute the values. They have given concentration of H plus in the question. You have to calculate concentration of OH minus. You will get it as 7.69 into 10 raised to minus 11, which is 7.7 into 10 raised to minus 11 molar, and the answer is option 3. Then IgM is a pentamer. Upon visualization of a pure IgM by electrophoresis on reducing polyacrylamide gel, the number of protein bands to be visible on the gel would be. So if the sample is reduced properly, then you can expect 3 bands which correspond to heavy chain, light chain and J chain. But the J chain band will be very light compared to other two, so it will not be clearly seen. So naturally you will visualize only 2 bands, that is of light chain and heavy chain. Answer is option 2. Then next one. A plot between phi and psi dihedral angles of a peptide bond is called Ramachandran plot. Answer is option 2. Then assertion, reason, questions. Assertion. Protein folding is essential for structure and function is still an unresolved problem in biology. Protein folding problem leads to aggregation and is the cause of many diseases. Yes, that is correct. So many prion diseases are caused by protein foldings. Then, so A is correct. Now reason. Lewinthal's paradox, that is for Lewenthal paradox is for protein folding, is a theoretical calculation of the time needed for a protein to fold. Yes. Accordingly, a polypeptide may take billions of years to fold if all the possible conformations are exercised during protein folding. So that is correct, and that is the correct explanation for A. So your answer here is both A and R are correct, and R is the correct explanation of A. Answer is option 1. Then next one, assertion. Prokaryotes have 3 RNA polymerases. No, prokaryotes have only 1 RNA polymerase, which is responsible for transcribing all types of RNA from DNA. Prokaryotic RNA polymerase 2, that is also not correct. Then reason. In eukaryotic RNA polymerase 2 synthesizes mRNA and is sensitive to low concentration of alpha amanitin. That is correct. So A is not correct, but R is correct. Answer is option 4. Then next one. Klinefelter syndrome have minor phenotypic anomalies with small testes and enlarged breasts These individuals have 47 chromosomes with 44 autosomes plus XXY. That's correct. Males with XYY syndrome have 2 Y chromosomes. Such individuals have antisocial behavior and aggressions. Their frequency is approximately 1 to 650. That is also correct, but it doesn't explain the assertion. So A and R are correct, but R is not the correct explanation of A. Answer is option 2. Then next one. Protein assertion. Protein kinases catalyze the transfer of gamma phosphoryl group of ATP to proteins. Serine, threonine or tyrosine are general amino acids that are phosphorylated. A is correct. Phosphorylation of proteins by protein kinases makes them aggregated and non-functional with subsequent degradation. That is not correct. Phosphorylation has various effects on protein activation, thereby increasing enzyme activity. Then localization, that is directing protein to specific cellular compartments. Then it can affect stability of proteins. Then it can inhibit protein activity. So R is not correct, but A is correct. So answer here is option 3. Then next one. While the pKa of free lysine is 10.5, the pKa of lysine decreased to 6.6 when present in the active site of acetoacetate decarboxylase. Okay, that's correct. The decreased pKa of lysine in the active site is due to the electrostatic effect of nearby positive charges. That is the correct explanation for A. So the answer here is both A and R are correct, and R is the correct explanation of A. Then next one, assertion. Sum of all proteins functioning in the cell is called the proteome. Yes. Proteomics is the systematic characterization of the proteome under a given condition. That's also correct. Reason. Expression of genome under a particular condition and its translation is the basis of cell's proteome. That is also correct. And it is the reason for A. So both A and R are correct, and R is the correct explanation of A. Answer is option 1. Then next one. Treatment of monosaccharides with sodium amalgam. So what is sodium amalgam? It is a chemical compound and it consists of sodium and mercury in their specific proportions, and it is a strong reducing agent, not an oxidizing agent. It is a strong reducing agent. So treatment of monosaccharides with sodium amalgam reduces their aldehyde and keto group to corresponding acid. Aldehyde and keto groups on reduction will not give acid, but they will give alcohols. So aldehydes will produce primary alcohol and keto groups will be reduced to secondary alcohol. So A is not correct. Then next one, reason. D-dulcitol and D-sorbitol are the corresponding alcohols of D-galactose and D-glucose respectively. That's correct. So A is not correct, but R is correct. Answer is option 4.